like to thank the organisers for giving me the opportunity to speak to you this evening. My name is Rebecca Wall and I'm a postdoctoral researcher within Alimentary Pharmabiotic Centre. The title of my talk this evening is Probiotics for a Good Brain Function. So APC was established in 2003 through funding from Science Foundation Ireland, and the mission of APC is to link Irish science with industry and society through excellence in research, education and outreach in gastrointestinal health. And one of our main objectives in APC is to investigate how the bacterial community that we have in our gut influence health and disease. And to do this research, we have 160 people, including microbiologists, immunologists, gastroenterologists and food scientists that are working on 6 main research teams. And these teams are led by 29 principal investigators. Staff is based in University College Cork and in Chiga's Morpark Food Research Centre. And APC has 2 significant industrial partners, Alimentary Health and GlaxoSmithKline. And I just want to bring to your attention that UCC was recently ranked 2nd in the world for probiotics research, and this is due entirely to publications from researchers in the APC. So we're looking at how the bacteria that we have in our gut influence our health. And in our gut, we have actually 10 times more bacteria than we have human cells in our body. And this bacterial community, or microbiota that we call it, is involved in many different important functions, such as immune processing, nutrient absorption and metabolism. And because of this huge metabolic activity that is associated with the microbiota, it has been referred to as a virtual organ. But it's also been shown that the microbiota is involved in disease processes that occurs outside the gut, such as obesity and non-alcoholic fatty liver disease. And one interesting finding that supports this is that germ-free mice, that is mice without a microbiota, they have 40% less body fat compared to normal mice. And this is even though that the germ-free mice consume more diet than the normal mice. So a lot of previous research have shown that microbiota can affect the quantity of fat in the host. But what we have seen in the research that I'm involved in is that it can also affect the quality of fat. Because we administered a Bifidobacterium breve probiotic strain that we had isolated from a baby to different animal species. And we then looked at how the administration of this particular probiotic strain would affect the fatty acid composition of different host tissues and organs. And what we noticed was that administration of this probiotic Bifidobacterium breve strain resulted in an increase in the beneficial omega-3 fatty acid DHA in the brain. And this was significantly higher compared to animals that did not receive this probiotic strain. And DHA is an important fatty acid for cognitive function, such as memory and learning. And the importance of this fatty acid in neurodevelopment has led to supplementation in infant formula. So this work was published this year in the American Journal of Clinical Nutrition, and it has also been featured on the front page of the Irish Times earlier on this year. I would like to thank some people in particular that have helped me with this research, and I also have a picture here of some of the people that are working within the Alimentary Pharmabiotic Centre. Thank you very much for your attention.